So after blotting techniques, the next important is PCR, polymerase chain reaction protein. So what is the procedure? Procedure is first we denature the sample. The denature and separate the sample at 95 degree centigrade. After separating, we just cool down. It is the heating temperature. We just cool down at 55 degree centigrade, which is cool down to promote annealing process. First we separate at going to the sample to very hot temperature, then we cool down at 55 degree temperature. Then we add primer. These primers, they go and anneal the target. The primers go and anneal the target. After adding primer, we also add the tag polymerase. Tag polymerases are actually the heat stable polymerases. So the tag polymerases are the heat stable polymerases, which these are the polymerases which are withstand with that heating temperature. And also we add nucleotides, double stranded DNA binding nucleotides. So now we separate the DNA segment or the sample, then we cool down to promote annealing, and after going to 55 degrees centigrade, this temperature, we just add all the ingredients which are necessary to promote replication. We are ready to replicate that that separated or denatured or unwind DNA strand in order to get the target protein. So for that we have the nucleotide, we have the primer, we have the tag heat stable polymerase. Now the replication process begin. So now at 75 degrees centigrade we reheat the sample, and which promote or elongate replication process. So at this by this cycle we just repeat the cycle, the heat and cool cycle, in order to get the amplify get that target protein. So whenever we get our target protein, we just keep on doing this heat and cool process in order to get that protein. So over here PCR is very much important in different aspects. First of all, what is the must known things regarding in order to start PCR? Before going to start PCR, we must know the flanking sites for primer making. We must know what are the flanking sites. In between the flanking sides, the target gene. So when we know the flanking site, we prepare a primer which go and anneal to the target gene. The next thing is PCR is used to amplify the target exon or the non-coding region. So PCR is used to target the exons or non-coding region. Then we have after this PCR is used to diagnose neonatal HIV or herpes encephalitis. PCR is very much highly sensitive or specific for diagnosing highly sensitive and specific for diagnosing toxogenic strains, but doesn't tell us about any active, does not distinguish or tell about active toxin production. And in this way, we just overly diagnose asymptomatic carrier. So this is pretty much a drawback for PCR. So in order to just confirm the process, let's review this. The PCR is used it is a lab procedure which is used to diagnose or amplify a desired fragment, which may be an exon, which may be a non-coding part, which may be a DNA, which may be a DNA. So we just amplify the target exons or a non-coding part It is used to diagnose neonatal HIV or herpes encephalitis, and it is very much sensitive for diagnosing toxogenic strain, but not distinguish about active toxin production. Overly diagnosed, this is a drawback. And also we must know the flanking sites in order to get this. So this is all about the polymerase chain reaction. We have also a reverse transcriptase type of polymerase reaction, but that reverse transcriptase type of reaction, it only detects messenger RNA level, which are complementary to our template. So they are in the first year of 2020, but not they are removed from 2021 edition. So there is a brief discussion regarding ELISA. ELISA, we have known that they are directly diagnose proteins. These proteins may be antigen and antibody, and it is highly sensitive and specific than Western blot, and ELISA is used for screen HIV. So whenever we want to screening test, we just do ELISA. So this is all about the important high yield aspects of genetic lab techniques.